Hi, my name is Marina Zapata, and with my partner Isabela Sepúlveda, we're gonna talk about how the electroacupuncture improves motor function after intracerebral hemorrhage through the pathway PPARY-EAAT2. We hope you enjoy and understand this presentation. Now, another topic that we're gonna talk about is how the electroacupuncture promotes the recovery of glutamate in the intracellular space. This is a schematic of how the electroacupuncture promotes the transcription of PPARY, which in turn increases the expression of EAAT2 and enhances the recovery of the glutamate in the intercellular space. For this, we're going to be using some techniques. The first one is the Western blotting. This process involves extracting proteins from the tissue samples and separating them by size using the gel electrophoresis. These proteins there are transferred to a membrane for easier detection. This membrane is incubated with antibodies, the primary antibody specific to the target protein, and a second antibody that is an enzyme or fluorescent tag for visualization. Finally, the protein quantity is measured by analyzing the intensity of the bands on the blood using specialized software. In the context of this study, the Western blotting is used to show that electroacupuncture can upregulate the expression of PPARY and EAAT2. We also have the quantitative PCR analysis. We're gonna do this to determine if the electroacupuncture affects the EAAT2 production in the brain after a stroke-like event, in this case the intracerebral hemorrhage. First, we're gonna do an RNA extraction. We're gonna convert that into a cDNA that is like a more stable form of cDNA. We're gonna amplify that with specific primers, and we're going to amplify the EAAT2 gene segment for detection. Then we're going to put that on a machine that quantifies the EAAT2 gene segment to assess activity, and we're going to compare that to a reference gene that is a B-actin for accuracy. With this quantitative PCR analysis, we're going to try to find, and it's going to help determine whether the electroacupuncture enhances the production of EAAT2, that is a protein that protects the brain after a stroke. For the results of the transmission electron microscopy images, we have a sham group that are the neurons that are healthy. They have many organelles and a healthy mitochondria. We have a model group that are the ones that suffered the intracerebral hemorrhage without the treatment. These mitochondria are swollen, damaged, and have broken membrane with fewer crystal. There are also fewer mitochondria overall. And we have the electroacupuncture group that were treated after the intracerebral hemorrhage. In this group, the mitochondria show less damage with some swelling but better integrity and higher numbers compared to the model group. In summary, the TEM images suggest that the electroacupuncture helps protect brain cells structure after a stroke-like injury, particularly by reducing the mitochondrial damage. We also have the results from the Western blot analysis of EAAT protein levels. We have a model group that is not treated with EAAT2 levels decreasing 3 days after the intracerebral hemorrhage compared to the sham group here. And the electroacupuncture group that were treated, here the treatment increases the EAAT2 protein levels after 7 days after the intracerebral hemorrhage. In summary, the Western blot analysis shows that the electroacupuncture helps the brain produce more EAAT2. That is a protein that clears excess of glutamate, so this reduces the potential damage after a stroke. We're gonna discuss the point of view of these three authors. First, we have the GBD 2019 stroke collaborators. They highlight the global burden of a stroke and they support the main authors by reinforcing the need for intracerebral hemorrhage treatment due to its global impact. Second, we have Kip, Hua, and Chi. They discusses intracerebral hemorrhage injury mechanism and therapies, and they support the main authors by providing a foundation for investigating secondary injury mechanism and potential treatment. Third, we have the Romera TEAI that identifies the EAAT2 as a neuroprotective PPARY target. They also support the main authors by confirming EAAT2 role in neuroprotection.